Welcome again to our lecture series on human physiology. This discussion is the first of a series of 3. They will be identified as ABC. Please note that I don't emphasize chapter numbers in case the textbook changes from edition to edition, just simply by altering the sequence of the topics by chapter numbers. So I like to present these as lectures, so this would be the A out of ABC, and I decided to split this A lecture into a 1 and a 2. The first part, the anatomy of the cells of the nervous system, the neurons, and the second part will focus on some of the membrane electrophysiological properties. So let's get started here. So about this chapter. So this first A-1 presentation is on the cells of the nervous system. Then the A-2 will focus a lot on the electrical signals in neurons. Then we'll proceed to the B and the C on cell-to-cell communication in the nervous system, then the integration of neural information transfer. So we begin with this brief overview of the organization of the nervous system. Recall that there's both anatomical and functional subdivisions of the nervous system. Anatomical, of course, being predominantly that contained within the cranial cavity and the vertebral canal, or the spinal canal, and containing, of course, the brain and the spinal cord. And the peripheral nervous system is anything outside of that, so where the cranial nerves attach. They are called nerves because we find them in the periphery. And then the spinal nerves, we find those in the periphery. So there's nerves and ganglia, collection of nerve cell bodies would be ganglia in the peripheral nervous system. And then tracts and nuclei, or gray matter, cortical matter, as you are familiar with in the cerebral cortex and the cerebellum in the CNS. So just kind of a quick review of some of that anatomical terminology. Now we'll just, in essence, consider it from the functional subdivision, that being represented by the enteric system here, the guts basically, where there's going to be, like the digestive tract, the visceral portion. And then over here we see sensory receptors that are predominantly associated with the body, or the soma of the organism, the somatic portion. So the somatic nervous system. As well, you can separate it in another way using different terminology, where the visceral is the involuntary nervous system. So we know that we're going to come back and think of the autonomic nervous system in the category of the involuntary, the visceral. And then the somatic being related to the voluntary nervous system. So we have voluntary, somatic, and involuntary, the visceral nervous systems. Now the reason that I'm introducing somatic and visceral at this point is because we will categorize receptors as we proceed through these series of lectures, and even the neurons, as you see here, a somatic motor neuron. We're going to combine them as having general, innervating the general structures of the body, like the skeletal muscle, as general somatic efferent neurons that would represent this functional pathway. Then again, the sensory receptor coming from a muscle, telling you how much stretch there is in it, would be triggering effects, triggering action potentials, a firing frequency in a general somatic afferent, those neurons bringing the affect to the integrating control center, the central nervous system made up of the brain and the spinal cord. So I know I jumped the gun on some of this, but I wanted to get that point across. So we'll be talking about general somatic afferents, general visceral afferents. They bring them to the same box here because we are kind of black boxing to get a basic circuit of input, integration, and output, which of course is the way that the organisms have evolved on this planet. Some type of external or internal environmental input that is integrated into a decision to generate an output as a response to that stimulus. So we also have a stimulus, integration, and a response system. So again, just quickly to review, we have an afferent, integration, and an efferent system, which is also an input, integration, and output system, which is also a stimulus, integration, and response system. So 3 ways of thinking about this on a simple level, and they kind of get integrated into a more sophisticated system as we proceed. So sensory system, the input on the left, and motor system, output on the right, and we'll have general somatic efferent, general visceral efferent. There will be of the cranial nerves some special visceral efferent, special visceral afferent, but in general there will be the general visceral efferent and general somatic afferent that we will concern ourselves with in this course. So when we talk about the special sensory structures, obviously there we're going to talk about special types of afferent and maybe efferent systems. So again, stimulus from the internal environment or external environment being transmitted to the internal environment, or the hollow portion of our gut tube is really the external environment, correct? The external environment extends from the opening of the urethra all the way up into the kidney, if you think of it that way. So there are these areas that seem to be inside our body that are actually lined by cells, surface cells or epithelial cells, containing a portion of the external environment inside the organism. So there's some type of signal. We have an input signal, some mechanism for that to be converted into a transmissible signal, action potentials, firing frequency within a neuron, and bringing it to the integrating central region, the central nervous system. And then there's obviously many types of signals that can come in through the somatic nervous system, the skin, the skeletal muscle, the subcutaneous tissue, and things like that. So we bring this information in to the CNS, and then some type of decision is made, reflexive, and then higher centers integrating the reflexive response, determining a patterned output. And patterned output is really important. If you don't pattern your output properly, you're not going to walk across the room. It's a good way to think about it. If there's a need for a somatic motor response, the general somatic efferent neurons are triggered in a patterned way for the skeletal muscles to bring about a tissue response. And if you walk across the room appropriately, of course, all the sensory input will help you to accomplish that so you don't fall over. You know when your knee is bent to a certain degree, or when your foot first makes contact with the floor, and you can start shifting your body weight from one leg to the other. So this feedback is important in integrating the continual patterned output. And then if there is a visceral component, the autonomic nervous system will be recruited appropriately and patterned. Don't ever think of the autonomic nervous system as either being on or off. There is always a basal tone, and you have the sympathetic and parasympathetic tones working against each other if they have a common innervation. So, for example, you have sympathetic innervation of the vasculature of skeletal muscle, but there's no parasympathetic innervation of the blood vessels in the skeletal muscle. So obviously, you're just talking about sympathetic tone there and local effects on the blood flow. But the gastrointestinal system, as you see it, should be getting to that. They kind of should have this arrow going over there, I think, and then back to the tissue response, but that's fine. They're showing some of the target cells here that are being controlled within the gastrointestinal system. Anyway, keep in mind that it's for those target organs or tissues or cells that have both sympathetic and parasympathetic input. Heart's a good one to think about. Heart rate is regulated moment to moment by the balance between these two. There may be cases where the tone is 0 for parasympathetic and something for sympathetic at basal levels, or vice versa. But still, it's the basal tone. There's always something. Typically, it's not 0. There's some low level of activity, like a 0.5 hertz firing rate of a sympathetic neuron is going to put a little bit of norepi onto the cardiac muscle tissue if you've got a lot of acetylcholine coming at 5 hertz to keep resting heart rate lower. Okay, so we have the target cells. So remember in talking about the endocrinology, the target cells are those that have the receptors for the chemical signals, and sympathetic is norepinephrine, parasympathetic is acetylcholine at the point where it's innervating the target cells. So the cardiac muscle, or the modified muscle cells at the SA node or AV node, as well as some of the atrial and ventricular cardiac muscle cells that might be targeted. If they express the receptors, they can respond to this input. So there is a control there. This would be a general visceral efferent to these structures. And then smooth muscle, exocrine gland cells, and then some endocrine gland cells can be targets, and then some adipose tissue can be targets as well to get a tissue response. And there will be some type of feedback through some type of sensory receptor somewhere. In some cases, like for the adipose tissue, it might be a peptide feedback that goes directly to receptors in the hypothalamus, which is studied in the neuroendocrinology portion in the prior series of lectures. Now let's add this other portion of the visceral nervous system, the enteric nervous system. And it can be considered separately. It's a very complex, sophisticated system, but in general, the motor part of it is associated with or included within the autonomic nervous system. But this is really considering the sensory components of this. You know that when your stomach is full, it's supposed to stretch and send signals to the central nervous system, mostly the hypothalamus, to say, I'm full, stop eating, right? So that's the reason for showing the enteric nervous system. But typically when you hear that term, people are associating it with the autonomic nervous system as well. So I know I spent a lot of time on that slide, but you need to have a global appreciation for this organization of the nervous system to appreciate and to more readily handle some of the integrative physiology that we ultimately will consider in the future, such as controlling heart rate, or what happens within this system when there is an imbalance of homeostasis, a disease, disorder, and pathology. Let's now then consider a model neuron to get a better feel, a review, as I know for you, or hopefully it's a review for you, of the anatomical structures of the cells of the nervous system. The functional cells, the functional unit cells, are the neurons. They are responsible for signal handling, that is, signal reception. As you see, the input of the signal at the dendrites, the integration among the dendrites and at the soma, and then all that can do is determine whether or not an action potential can be fired, and then how frequently the action potentials will be fired in a block of time, and then how long it will be before another set of action potentials are generated, such that they can be conducted down the axon for the long-range, long-distance signaling to the terminals, where, of course, the digital action potential at a certain frequency and interburst interval. So if you burst here at the axon hillock, 3 action potentials, that's in 1 second, that's 3 hertz, correct? And then you have a 20 millisecond interburst interval and then send 3 more, you're sustaining a 3 hertz with an interburst interval of, what did I say, 30 milliseconds, something like that. So hopefully you're seeing that the 2 properties of the coded signal, the output signal that is digital, are the frequency and the inter-signal interval of time. So that's the coded signal that can be sent down. And then, as we'll see when we get to Lecture C, but as you should know, there is an exocytotic event here, and we will see that in the upcoming slide. So one action potential is going to be associated with a certain number of vesicles dumping a certain amount of chemical into the cleft, and we discussed that briefly in the neuroendocrinology section. So again, we have an input, integration, and output. So at the functional unit level, at the cellular level, we have the same type of a circuit system or functional system as we have globally for the nervous system collectively, as we saw in the previous slide. So the simple, the most reduced system is the same as the full global system. So I don't know what would happen if this worked differently. I don't know how it would work differently, but anyway, if this worked differently than the global system, we might not be very intelligent organisms, huh? Very functional at getting around in the environment. So to come back then, of course, there's nucleus and all the subcellular organelles, as you reviewed in the review lectures. This first portion of the output process, so the dendrites are the processes of the cell body or soma that are receptive for the input signal, and they contribute to integration. And then the attachment of the output portion or process extension of the cell body is the axon hillock, and that is the anatomical term for it. The functional term for this area is going to be the spike zone. So this is functionally where the voltage-gated sodium channels are at a high enough concentration such that an action potential can be generated. So again, we keep in mind anatomical and functional terminology. The same location, the axon hillock, but a different term, the spike zone, because we're now referring to it in the functional context. And we know that the conduction is going to be impacted by many parameters, the diameter of the axon, and whether or not it is myelinated, as you certainly should be aware. Now let's just take a moment to look at the point of communication between the neurons. So an interneuronal type of a chemical signaling. The synapse is actually this entire structure, the junction between the neurons, so an interneuronal junction. And we know that there's different types of synapses, as we will see when we proceed. So there is a presynaptic axon terminal. So this whole cell, with reference to this synapse, this entire neuron is presynaptic, and this entire neuron is postsynaptic. So you see that indicated here. So this whole neuron is postsynaptic with reference to that synapse. You come back here to a synapse here, and then this one becomes postsynaptic, and that one becomes presynaptic. So that's the circuitry part of this. So there is a space, a gap between them. That's the synaptic cleft. And then there is a presynaptic axon terminal. It's going to have a membrane and a postsynaptic site. In this case, it's the dendrite, because this is an axodendritic synapse. And there's going to be a presynaptic membrane and a postsynaptic membrane that are highly specialized for the transmission of the signal. And remember what we said was that the coded signal, the digital signal of the action potentials with a frequency and an interpulse or interburst interval is the coded signal. It gets converted to an analog signal, the continuous range of concentrations that are possible for the transmitter, the chemical messenger, within the synaptic cleft. So the synaptic cleft concentration of the transmitter is the most important aspect of this whole functional circuit between these two neurons. Well, now that we know that there's the cell body, the processes, dendrites are the receptive and axons are the conducting portion, we can point out the exception, but then use that to talk that information to talk about the anatomical and the functional categories of neurons. So we can think then of sensory neurons, and that would be the functional classification. And then the anatomical classification with those that would look like they have only one process coming off them, but that branches into two of them. One that goes to the periphery. Remember, this is sensory, so this would be a somatic sensory neuron, the primary somatic sensory neuron, going from your fingertip to the dorsal root ganglion of the spinal nerve close to the spinal cord, so that's still peripheral. And then the central process, did I call that a peripheral process? Then the central process that goes into the spinal cord, so this would be in the CNS, and this would be in the PNS. And the reason we call this a dendrite is because it carries information toward the cell body, and we call this an axon because it carries information away from the cell body. However, if you look at the molecular biology of this peripheral process of the pseudo-unipolar primary sensory neuron, functionally it has the capability of generating action potentials, so functionally it's really an axon. But you have a receptive portion most distally, so this is a unique anatomical arrangement. And functionally, it simply makes sense to carry somatic sensory information over long distances, from your tip of your toe all the way to the spinal cord. It's a long distance. You want to myelinate it and use action potentials to get the information there. For more highly specialized sensory structures, a simple bipolar cell anatomically can achieve this task, because this dendritic portion, and indeed it is a dendrite, is covering a much shorter distance. And then the axon will carry the information into the CNS. So when we talk about the special senses, this will be involved. So this obviously is going to be associated with a special somatic afferents, or special visceral afferent type of sensory modality systems, and this is going to be associated with general somatic or general visceral efferent systems. So to throw those terms in at this time. So example, smell and vision. So smell is going to be special somatic, Vision is special somatic because it is a biophysical property, right, the photon of light. And then smell, olfaction, everything is dissolved within an aqueous medium, so it's mucousy and kind of visceral-like, right? So it's called a special visceral afferent for the sense of smell, for olfaction. And then special somatic for vision, because you're going to have the photoreceptors providing the ultimate signal to the dendrite for this to bring the visual information through the optic nerve into the brain. Continuing with this then, I'll try to spend less time on each one, but it's very important to make those points at this time for you to understand neurology in the context of physiology. So then we can look at different types of interneurons. So we have sensory for the input, integration that we're seeing here, or interneurons. And then obviously the next that we're going to look at is going to be the efferent, or the output neurons. So these functional categories of the afferent to bring it in, integration, or interneurons to integrate, and then of course the efferent, the motor neurons, to take it out of the black box system that we talked about on that first slide. So obviously these are not bipolar or pseudo-unipolar. They have multiple processes coming off them. This is the classic Purkinje neuron of the cerebellum that we see in the first slide. We see the cross-section of the cerebellum in the first slide of each presentation, where these cells, their dendritic trees are, and it's even on the cover of the textbook, at least this edition. These dendritic trees are all green, and then the soma is yellow with the nucleus being red, and then in many cases you can see the axon's green color extending away from it. So it's really a beautiful color illustration of this type of very complex neurons. So there's a lot of integration that can go on here. They're not showing the alpha motor neuron, the pyramidal neurons of the cerebral cortex, but they as well have a very sophisticated and extensive dendritic tree, all attached to a single soma. So there's a lot of integration that must occur out here before information gets to the soma, but it doesn't matter what all this is doing. There's only two things that it can affect at the trigger zone, located at the axon hillock. That is, first of all, whether or not an action potential will be generated, but how many within a single burst or pulse, and then how long before the next pulse is going to be sent, and is that subsequent pulse going to be the same number of action potentials as in the previous one? So can you get this neuron up to 100 hertz, sending a pulse of 100 action potentials every 3 milliseconds or something? So that's a very high firing rate, probably in seizure if that's happening. But anyway, I want to continue to emphasize that no matter how sophisticated the neurons are, really only once it's determined that an action potential can be generated, there's only two more decisions that have to be made. The firing rate, how many per second, and the interval between the next between that burst and the next burst or pulse. Excuse me. So then we see these are multipolar neurons. Obviously, they have many poles. And this one, the N-axonic, and simply because they can't see the axon, I'm not going to say anything about it whatsoever. My experience is that there's an axon somewhere, it just wasn't seen. Anyway, it may turn out to be something different in the future. At present, it's been added. I don't recall ever seeing it in a textbook before, the N-axonic. It's simply because they can't find the axon. So who knows what it's doing. But predominantly, we see all the other kinds of neurons. And the efferent is the classical multipolar neuron, such as the alpha motor neuron that innervates the skeletal muscle fiber directly. So you have this extensive dendritic tree and the large cell body and the axon that is heavily myelinated with these multiple terminals. This is called the telodendria, as it branches multiple times to innervate several different skeletal muscle fibers, for example, to form a motor unit, as we will see. But this would be the same appearance of a general visceral efferent motor neuron coming from the spinal cord as part of the sympathetic portion of the autonomic nervous system to innervate its postganglionic neuron somewhere out in the periphery, like the sympathetic chain alongside the vertebral column. And obviously we're going to talk about the autonomic nervous system in a separate set of lectures. So the cells of the nervous system then, let's think of the axon transport system. So axons will be the site where we see a tremendous amount of transport because you have these long distances. Again, let's switch from the sensory neuron from the tip of the toe to a motor neuron going from the spinal cord all the way through the nerves down to muscles in your foot. So a tremendous distance, and this cell body with its genes and all of its cellular mechanisms for proteins and membrane lipids and all that other type of essential component, cellular component system that must be maintained down here at the very tip, at the muscle in the bottom of your foot, has to be accomplished. And so there's got to be stuff going back saying we don't need this anymore, and then saying, well, then we need some new stuff. So constantly things going back and forth, and some slow and some fast. So the slow axonal transport moves material by axoplasmic or cytoplasmic flow, kind of a bulk flow type of a system, 0.2 to 2.5 millimeters per day. So picture your 10 centimeter ruler and the single centimeter that is divided into millimeters, and 2 or 2.5 of those, 0.2 or 2.5 of those, that distance over a day, and how many of those are between your your lower lumbar spinal cord at the bottom of your rib cage, all the way down to the bottom of your foot. So the cytoplasmic is pretty slow. The fast axonal transport moves organelles at rates up to 400 millimeters a day, so it can get it there very quickly. That's like for the critical neurotransmitter sustaining systems, or presynaptic membrane sustaining systems, and the ion channels and those types of things. So there is a forward movement, or anterograde transport from the cell body to the axon terminal, and then there's one that brings it back, the retrograde, transport from the axon terminal to the cell body. We can discuss many of these issues with this illustration, and I really like the way that they put this together, because they have what I think is one of the most complete that I've seen in a textbook in a long time. So I really do like this illustration. So we are considering the fast axonal transport of the membranous organelles, but also in this context we can review the membrane recycling system. So we recall that the messenger RNA comes from the DNA in the nucleus and passes through the nucleus once it is processed to meet up with ribosomes for protein synthesis, and we need to get certain proteins to the axon terminal, but also inserted into the dendritic membranes, either here or way out at a synapse, far out. You saw recall the Purkinje cells of the cerebellum. These the proteins that are produced and packaged in membranes in the vesicles here have to travel 100 micrometers or 200 micrometers to get out to the dendrites, the distal dendrites, to maintain structures there. There's some of these mechanisms, these subcellular subcellular organelles, that also become resident out in the distal dendrites to help overcome that challenge. So the messenger RNA needs to get out there, but still we have that type of a transport. But every place you have membrane being inserted, you've got to have nearby membrane coming out. If you don't remove membrane from a cell, it just continues to grow and grow and grow as you insert it in there, right? So there has to be a recycling. So here they're showing coming from the ER to the transport vesicles to the Golgi, as you should know, with lysosomes, primary lysosomes being produced, or transport vesicles. These would be secretory or synaptic vesicles, with the anterograde transport along the microtubule system to the axon terminal. So here, for example, we're seeing peptides synthesized and packaged for, say, a neurohormone. Then number 2, the fast axonal transport along the microtubule network. And then the vesicle contents released by exocytosis here. So if you insert membrane here, you have to bring membrane back in somewhere else. So you see the synaptic vesicle recycling over here, or membrane recycling, some of which is old, and there's other debris that needs to be returned to the soma where the lysosomes are for the old membrane components to be digested in the lysosomes. And this is part of the faulty system. As we age, we accumulate in our brain cells oxidized lipids that cannot be completely degraded and recycled. So it's very important to understand all of these mechanisms. As well, they're not showing it, but anywhere nearby there has to be endocytotic vesicles pulling membrane in to maintain the membrane of the soma and the dendrites. So this is across the entire surface of the neuron that we have to have this membrane recycling. But for vesicles, this is very important. It is possible to exhaust the reserve of vesicles in synaptic terminals, as happens with certain drugs of abuse, such as ecstasy, the MDMA. It will initially cause just a burst of exocytosis and an exhaustion of the vesicles, so the rate of recycling and regenerating vesicles is overwhelmed by the rate of exocytotic fusion. So you get swelling of the terminals and less serotonin to be available the following days. So after a weekend of ecstasy, people don't feel so good because there's, the vesicles haven't been, the terminals haven't recovered from the toxic effects. So I hope that helps you to understand this. And then, of course, you have this movement. So and it's not just set up this way on the top of the axon; it's going that way, and this, they're going back and forth. And there's many of these microtubules in there. So there's a lot of vesicles going this way, and a lot of vesicles going this way, using ATP for the proteins that are associated. There's kinesin and dynein. For example, some of the motor proteins responsible for this, and ATP is expended. So because you have so many, and it's just a three-dimensional complex going that way and this way, so like when you walk across a pond, you bring water with you from one side to the other, in essence, and if there was a whole bunch of people walking from one side to the other simultaneously, back and forth, they're going to move water with them, and that contributes to the slow transport, the bulk flow of the fluid. So the bulk flow, the slow transport, doesn't happen just all on its own. It's in association with the utilization of chemical energy to produce the kinetic movement of molecules. And in that case, it's large-scale biological macromolecules, the vesicles. So the cells of the CNS continuing, now we think of the supportive cells, the glial cells, and their functions. So they're essentially the two cell types in the nervous system, the functional unit, the neurons, and the supportive cells, the glial cells, or those that glue everything together. The glia means glue. So glial cells are found in the peripheral nervous system and in the central nervous system, as we'll see in the next slide. And in each location, they provide physical and biochemical support for the neurons. So the anatomical part and the functional part, as we are considering it here in this functional anatomy course or physiology course, right? So the peripheral nervous system contains the cells that surround the neuron cell bodies, wherever you might find them, in a ganglion. A collection of nerve cell bodies in the peripheral nervous system is a ganglion. So the dorsal root ganglion would be a somatic sensory ganglion. And remember that pseudo-unipolar neuron, the cell body, hanging off the peripheral and central processes, it has to be surrounded and supported, and it's satellite cells that will surround it out there. So they support the cell bodies in the ganglia. And autonomic ganglia would be the same thing. The postganglionic sympathetic neurons in the sympathetic chain are going to be, their soma and dendrites are going to be surrounded by the satellite cells. And then in the peripheral nervous system, those cells that wrap around either once or multiple times the axons in the peripheral nervous system are called Schwann cells. So if they wrap around the axon multiple times, then they are forming the myelin sheaths. But every axon will be surrounded by that, and we'll see it in an upcoming illustration. At least one time they will be pushed into and embedded in just one of these. So if you took away these multiple layers here and put an axon in there, but it was just one layer of membrane that came in and around it and then down like that, kind of like having a long cylindrical balloon, and you take a pencil and you just push it into it once, but you don't rotate it to wrap the balloon around it. You just push it into it until the balloon closes off on the outside. That would be an unmyelinated axon, but still it is surrounded by, supported by, physically and biochemically, a Schwann cell in the peripheral nervous system. So remember, axons don't just hang around floating in the interstitial space. There's no surface area of a neuron along any of its length that is uncovered. There's some type of glial cell very, very close by to it. If not, what do you have? You have an interneuronal junction, right? Or a neuromuscular junction, a neuroeffector junction, right? Now these Schwann cells are supportive. They contribute neurotrophic factors. They express the genes for sets of or subsets of neurotrophic factors, and they secrete them into the interstitial fluid surrounding the axon that they are surrounding, either once with one wrap or with multiple wraps, myelinated, to keep the neuron, the axons alive. So that was an important discovery where we finally began to understand better how the Schwann cells that are either supporting non-myelinated or, very importantly, myelinated axons help to keep the axons alive. Remember, we said it's quite a task for the cell body of a neuron to keep the axon alive down near the foot, while the Schwann cells assist in that. So the cells of the nervous system, we continue with the glial cells, where they are found in the central nervous system now. And I wish they had pushed this microglial cell over to the side, because we then would have this arranged such that we have the three central glial cells that are of neural primordial cell origin, ectodermal or neuroectodermal origin, as it would be called, or neuroepithelial cells, and then one that is from a peripheral origin, a mesodermal type of a cell, if you recall any of your embryology. So these microglia are the modified immune cells. They actually migrate into the central nervous system from the blood through the vascular wall. And then they differentiate into these types of modified immune cells. They are phagocytic cells. They're scavengers, okay? So the oligos are going to be the analogous cell to the Schwann cell of the periphery. However, we'll distinguish that. You can see right now that you have one oligo, in this case, that is myelinating one internodal segment on two different axons. We'll expand on that in a moment. So the oligos are the ones responsible for forming the myelin sheaths. But what if it wraps around only once? Well, they're also there surrounding one time, just the same balloon thing. Picture this as just one, not multiple wraps as you see it cut here, but just a single one around a pencil, a portion of a pencil. Then that would be an unmyelinated axon. Either way, the oligo is there in the CNS to act as the Schwann cell in the PNS. So each myelinated segment of an axon, or internodal segment, as you might know, it's called the node of Ranvier. We'll see that when we talk about conductance. Each internodal segment in the peripheral nervous system has one cell there, and therefore you'd find its nucleus, a Schwann cell. In the CNS, you will not find the nucleus at that location because a cytoplasmic projection is connecting the nucleus, the soma, if you will, containing the nucleus, with that myelinated segment of an axon. So these oligos are more sophisticated. They're capable of maintaining more portions of single axons or portions of more axons. And then the astrocytes are taking care of predominantly everything else. They provide substrates for ATP production. They help form the blood-brain barrier. They secrete neurotrophic factors. They take up potassium, water, and neurotransmitters. So they are attending to the interstitial portion of the extracellular fluid compartment surrounding the neurons. And they are thought to be a source of some neurostem cells, as well as the cells lining the cerebroventricular system, called the ependymal cells. And the ependymal cells create a barrier between different compartments, such as the cerebrospinal fluid and the interstitial fluid surrounding the other cells of the nervous system, astrocytes, microglia, oligos, and the neurons. So here we get to take a look at that more complete picture. I really like this picture, and we're going to spend a few minutes on it to take a look at nearly everything that's illustrated here. So what we are looking at, we see on the right hand side here, is a cross section of the lower cervical spinal cord. That would be C1, C2, C3, C4, C5, C6, C7. So that's the cervical spinal cord, and then it begins the thoracic there. That cervical dilation should be shifted up a bit, but don't worry about it too much. So in that cross-section, we're aware that the white matter, the myelinated tracts, are on the periphery, and the gray matter is the more centralized portion. And we're just going to take a closer look at the gray matter around the central canal, as it's called, and some of the nearby white matter, to look at how these cells that we just talked about of the CNS would be illustratively arranged and organized. So we follow this top right corner, the central canal, up here to the top right, showing the ependymal cells lining this fluid-filled cavity that runs throughout the length of the core, or the central part of the spinal cord. The central canal is filled with cerebrospinal fluid. As you know, between the arachnoid and the pia mater is the subarachnoid space, where you also have cerebrospinal fluid on the outside of the spinal cord. The volume of that cerebrospinal fluid is obviously much more than you find just in this very centralized canal. But anyway, there is fluid there. And it is extracellular fluid for exchange it's kind of the lymph fluid, as you recall, of the CNS. There's no lymphatic vessels. There are blood vessels. Here we see a blood vessel running through this area. There would be many blood vessels. They're just showing one. And the the astrocyte foot processes completely cover nearly every angstrom of the what's called the abluminal surface of the blood vessel. So away from the lumen of the blood vessel. If you did a cross section here and looked in, you'd see the lumen, right? And the adluminal would be adjacent to it. So the lining of the endothelial cell, then the abluminal would be the farthest away. Then there's a pericapillary space, and then you'd have this continuous containment by these foot processes of the astrocytes. So many astrocytes, just wherever they have a process that encounters a blood vessel, they cover some of the surface of it. The cytoplasm flattens out in development, obviously, or if there's a revascularization, to cover it, because they are the guard keepers, the gatekeepers, the guards, the blood-brain barrier, saying you can come in, you can go out, and they are very dedicated to maintaining an appropriate electrolyte environment, because if you screw that up in the interstitial space surrounding neurons, you're dead, right? Your neurons don't work, you're not going to live long. So these ependymal cells are associated with contributing to the removal of substances that you typically might think of would end up in lymph and peripheral tissues. They form a physical barrier for any leakage, so it is only through some type of permissive transport system that they allow things to move from the CSF into the interstitial fluid, or vice versa. And you see also that they have contact with some blood vessels, more heavily in some areas than in others, like the neuroendocrine hypothalamus. This is a very important feature of the ependymal cells there. So that takes care of the ependymal cells. We saw one feature of the astrocytes. There's actually two types of astrocytes. Those that are in the gray matter are nice and plump, lots of cytoplasm and all their projections going out, and they cover every surface of, they're just showing a few of them on here, but there would be no blue exposed. Just as it's covered like this, these would be covered. There's no portion of a neuron that is not covered by some type of glial cell. Let's make that point right here, where an oligo is myelinating this internodal segment. This is called a node of Ranvier. You see the term down here in the bottom left, node. So in between the nodes, well, there's the axon hillock. It's still an internodal segment. But where the oligo ends, you have astrocytes, and not just one covering it there, all the way around it, just as you picture around this capillary here. So there is, if it isn't a neuron, it's a glial cell that you see in the tissue. So these guys are nice and plump. They're called protoplasmic astrocytes. And then in the white matter, they dedicate all the cytoplasmic processes they can to the protecting the cells that are there exposed at the nodes, so they don't need a lot of plump cytoplasm to do that, because mostly the oligos are covering the neurons. So these become what look like fibrous types of cells, so they're called fibrous astrocytes. But astrocytes are in both the gray and the white matter. Microglia are going to be pretty much everywhere. They're just showing it here in the gray matter. And then we see the oligos. So anywhere you have an axon terminal forming a synapse, you do have exposed neuronal membranes, but it's one neuron to another. In any place otherwise, and they're just showing illustratively one here, but all the way around, and all of this blue, none of it would be seen. It is all completely covered with the, let me just say zillions, to be kind of ridiculous about it, but the zillions of other astrocytes and their protoplasmic processes covering all of this. So these little things here are not synapses. Those are astrocyte processes that are dedicated to maintaining the interstitial space for these other cells' survival, such as the oligo here and the neurons there, anywhere along their length. And then these are the synapses. So we'll see another illustration of how many synapses are covering the surface of a neuron. So a lot is left off so we can see the blue neuron here. But if you were really floating within a spinal column, you wouldn't see that in the gray matter. Of course, in the white matter, you have the myelinated and unmyelinated axons. They're not really showing an unmyelinated axon here really very well, but they will be there as well. but mostly you're going to have these very heavily myelinated axons within the tracts of the spinal cord. And then we see the oligos forming the multiple wraps for the myelinated segment of the myelinated axons. So I think we covered everything very nicely on that. Now let's think about how a Schwann cell would form myelin. So we begin with this image, and if we just stop here, this would be how an unmyelinated axon would appear. But one Schwann cell can have numerous of these guys pushed into them. So one Schwann cell can have multiple unmyelinated axons pushed into its cytoplasm to tend to them. But if it's going to form myelin, then it will continue to form membranous wraps around the axon as it's forming. And in us right now, our myelin is pretty static. The turnover is not very high at all. This would not be an ongoing process to this degree, but in a young child it would be. If you had an injury to peripheral nerve and you were having regeneration, fortunate enough to have regeneration, this would occur again as a repair process. It would be as perfect as the first time when you were from birth to when you could walk and talk and do everything you're supposed to. Maybe, maybe not, but you would have some degree of functional recovery. So initially, the Schwann cell wraps around the axon. They're saying many times here. The first time, let's say, and then it continues that process, and the Schwann cell nucleus is pushed to the outside of the myelin sheath. So this thick layer of lipid, remember the lipid bilayer membrane is what's going on here, and it's folding upon itself, and the cytoplasm, except for a very little bit, gets squeezed out. And so this is a lot of lipid wrapped around it, and that myelin consists of multiple layers of the cell membrane, therefore it is a lot of lipid. So the myelin formation in the peripheral nervous system is illustrated in that photograph. So then again, we see the Schwann cell being very convenient for us to recognize myelination as an anatomical feature, and then later we'll talk about the functional relevance of this organization. So about 1 to 1.5 millimeter length for the internodal segment that is myelinated, and very likely this length would be similar in the CNS. To tell you the truth and be very honest, I don't know exactly, but I would presume it to be very similar. However, if we were to look at an internodal segment that is myelinated in the CNS, we would not see the nucleus. So this is illustrating the peripherally located nucleus that got squeezed to the side during the myelination process. So the Schwann cell nucleus, as we said, is pushed to the outside, and then the multiple wrapped layers are called the myelin sheath. And the myelin consists of the multiple layers of this the cell membrane, and repeating themselves there. And so we had talked about the node of Ranvier is a section of the unmyelinated axon membrane between two Schwann cells, and therefore I utilize the term of an internodal segment to talk about where the myelin is actually located. In the periphery, satellite cells are going to be all around here, just as astrocytes were around all of these structures in the CNS. So when I first talked about the satellite cells, I said they surround every portion of the soma and the dendrites and a ganglion. Well, you're also going to find them everywhere within a nerve to maintain the external environment, with reference to the inside of the axon, the extracellular or interstitial fluid portion of the extracellular fluid compartment right around these nodes. And they also are there to help out the Schwann cells. So this finishes our first presentation on this 8A.